All right, sorry about that, guys. We'll come back for part two. I had someone pop into the office. So we are going into our actual components of our nephron. So we just talked about the renal corpuscle that contains our glomerulus and the Bowman's capsule. And so that's how we get our filtrate. And so the proximal convoluted tubule is going to be where our filtrate first passes. So its main job is to drain and kind of hold the filtrate that it's getting from Bowman's capsule. Our loop of Henle has both a descending and ascending portion. And on the different portions, descending versus ascending, we're going to have water and solutes pass through these very thin walls. So just like a capillary, it's super thin to allow for ease of passage of material. And so things are typically going to diffuse from high to low concentration. So most of it is passive. Our distal convoluted tubule is after our loop of Henle and prior to our collecting duct. And the collecting duct themselves are what are going to actually empty into the calyces. Remember, we had our renal pyramid, papilla, and then the calyx. And so our calyces end up at the renal pelvis. So this is how we're going to drain the urine. And so this is what's kind of carrying fluid from that outer cortex region of the nephron into the medulla. So this order you definitely need to know. So this is the flow of filtrate through the nephron. So this is our renal corpuscle we started with. That's how we're forming our filtrate. The glomerular capillaries are under high pressure. Fluid is pushed out. The Bowman's capsule collects that and sends it to our proximal convoluted tubule. We then have the descending and ascending loops of Henle. We're going to have water and other solutes moving both in and out of our filtrate during this step. The distal convoluted tubule is going to collect that filtrate from the ascending loop, and it's going to be the bridge to our collecting duct. The collecting duct then filters to the papillary duct, where we end up meeting our calyx and then eventually the renal pelvis. Now, blood flow through the kidney, we're not going to talk about quite as much. This shows our order. So this is something that you should at least review and be familiar with, but I don't necessarily expect you guys to memorize this and be able to put it in order. So we have our renal artery that is initially supplying the blood to the kidney. Our renal artery is going to branch into an interlobar artery. We then have arcuate arteries following interlobular. So interlobular is farther down the chain. We have the afferent arteriole. So remember, afferent arrives. So this is how blood is going to enter the glomerulus. Blood is going to exit the glomerulus or that capillary network via our efferent arteriole. So remember, efferent exits. We then have the peritubular capillaries, the vasa recta, and so those are part of this network. Let me see. So our vasa recta and the peritubular capillaries are kind of around our two ducts here. And then we're going to go into the veins. So we're all arteries up until this point here. Our peritubular capillaries and the vasa recta are kind of our bridge to the veins. So then we have an interlobular vein, just like we had an interlobular artery, the arcuate vein, like we had an arcuate artery, and then the interlobar vein. And if we had a number 13 here, we would be at the renal vein. This puts it in order. So 1 through 13. So this is a good way to practice kind of how that blood flow happens, and it shows what happens. So we have our renal artery. That's how a majority of the blood is getting in. And then our interlobar arteries are going to be present in each section here. So they branch to an arcuate artery, which kind of wraps around that medulla portion. And then our interlobular artery extends off of that and will eventually become an afferent arteriole for our glomerular capillary network. And then you kind of continue to follow that path. Okay, urine formation itself is definitely important to know. So it's really three primary processes. We have filtration. We said that occurs in our renal corpuscle. So we have the fluid portion of blood, that blood plasma that is pushed out of the glomerulus and enters our Bowman's space. Bowman's capsule collects that fluid and sends it to the proximal convoluted tubule. We have tubular reabsorption. So this is where we are going to remove substances from the filtrate. So remember, our filtrate is what we just made. This is going to go from the filtrate back to the blood. So we're reabsorbing them. We already absorbed them once into the body. We then push them into the filtrate. Now we're going to reabsorb. So most of the solutes that are pushed out of the glomerular capillaries in filtration, most of that stuff we can use in the body. So we're going to reabsorb it. And then finally, we have secretion. And so most of this is done by the distal convoluted tubule. I should backtrack here and say our proximal convoluted tubule in our loop of Henle is primarily where reabsorption happens. Our distal convoluted tubule is where we tend to secrete a majority of things. There is a small amount of reabsorption that still happens here. But the secretion aspect is when we're going to have substances that are still in the blood at our nephron, and we're going to put that into our tubule. So to go through that again, we have filtration in the renal corpuscle. Within the proximal convoluted tubule in our loop of Henle, we are going to put a lot of these solutes back from the filtrate into the blood. And then in our distal convoluted tubule, we end up with a little bit more secretion. So stuff that's still in the blood, we're going to now put back into that tubule so that we can get rid of it. So we'll look at these steps a little bit more in detail. So our first step of filtration, so we get water, ions, and molecules that are small enough to pass through our glomerular capillaries that end up in Bowman's capsule. So about 19% of plasma, so about 1/5 of the plasma or fluid portion of the blood is going to become filtrate. Only about 1% of that is going to become urine. So that means 18% of it is going to be reabsorbed. So like I said, a lot of that filtrate is reabsorbed. Only small molecules are going to pass through our filtration membrane. So that's going to be really tiny stuff like sodium, potassium, little ions, some really small proteins, mostly water. So filtrate is going to depend on filtration pressure. So in a sense, the more filtration pressure there is, the more filtrate we are going to make. And our body can somewhat control the amount of blood that is being introduced into that renal corpuscle to help control this rate. So there's a few things that also influence this rate outside of blood flow, and that has to do with pressures of fluid on either side of the membrane, and then osmotic pressure as well. So we have differences in the concentration of water that's going to influence how much fluid is moving. So we're not going to get into detail on this. Just know that these are a couple factors that affect how much filtrate we are going to make. So reabsorption, 99% of that filtrate is reabsorbed. So like I said, we push out a lot more stuff than we really want to get rid of. So we're going to reabsorb a lot of our ions, nutrients, water, anything that we can utilize for ourselves. So we're going to pull in a lot of that. We only want to get rid of a small amount of filtrate. In the descending loop of Henle, this is where our filtrate concentrates. So in the descending loop, this is where the water that's going to be reabsorbed primarily is done. So now instead of a watery filtrate, we mostly have what we want to get rid of. This reabsorption continues at the distal convoluted tubule, and then eventually we'll reach the collecting duct, and all of this is controlled by various hormones. So we're not going to get into those hormones very in depth for this lecture. Secretion itself is when we're going to remove some additional substances from the blood. Most of these are byproducts of our metabolism. So remember we said we use oxygen and glucose to make ATP. One of the byproducts we talked about was carbon dioxide, but we have a few other byproducts like uric acid, urea, creatine that are also going to be included. This is also going to include high concentrations of different drugs or medications that we have in our bodies. And so some tubular secretion can occur passively. So if we have a high concentration in one area and a low concentration in another area, we can have normal diffusion occur. But if we don't have that concentration gradient to drive the movement that we want, then it's going to take energy to move stuff from the blood into the tube. Remember that at this point, we're in our distal convoluted tubule. So getting away from the nephron itself a little bit, now we're at the ureters, and these are the small tubes that are going to carry urine from the renal pelvis of our kidney to the actual bladder. Our bladder is down in the pelvic cavity, and its job is to store urine, and it can store a lot of urine. A couple thousand milliliters or 1,000 milliliters is what we consider the max, which is a lot of fluid. So kidneys down into ureters. The ureters are made of this kind of connective tissue and smooth muscle layer known as transitional epithelium. So we have this connective tissue on the outside, smooth muscle on the inside. The urethra is going to carry our urine from bladder to external environment. That's really its only job. Just like the rectum, we have an internal and external sphincter. So our internal urethral sphincter is smooth muscle, so that means it's involuntary. And this is at the junction of our urinary bladder to the urethra. And so it's going to prevent urine from entering the urethra. Our external urethral sphincter is skeletal muscle, meaning it is voluntary. So the term micturition is kind of our scientific term for urination, and this happens via the micturition reflex. And so this is activated by the stretch of our bladder wall. So when our bladder stretches to a certain point, it's going to increase the conduction of action potentials, and that is going from bladder to our spinal cord. So it's not even going up to the brain yet. So when the bladder stretches to a certain point, we get a kind of a signal sent to the spinal cord that we have a full bladder. We're now going to get increased parasympathetic input. Remember, that's rest and digest. So parasympathetic action potentials will cause the smooth muscle of the bladder to contract. So that's how we kind of push the urine out. And that stretch reflex also stimulates sensory neurons in the brain that gives us that feeling of the need to urinate. So just to be clear, the reflex itself is started by the bladder and runs through the spinal cord. The only thing in terms of micturition that runs through the brain is the actual sensation of the need to urinate. And this just gives you a little diagram of that loop. So bladder stretches, we get action potentials conducted to our spinal cord that causes the parasympathetic nerves there to increase their input. That's going to cause contraction of the bladder. At the same time, our brain is going to get the signal of the sensory aspect of the need to urinate, and we can voluntarily control our external urethral sphincter to start and stop the flow of urine. And that covers everything for chapter 18. There are a couple of hormones that I'm going to introduce to you guys that controls this reabsorption aspect of things somewhat. But I would like to kind of put it on paper for you a little better so that You're not just listening to me speak. So you'll see here shortly, you'll get a diagram, kind of little chart to help you match up some hormones that are relevant in this chapter.